Okay, welcome to a new SEM webinar. Today we have a seminar from the Chemical Biology group that is coordinated by Sonsoles Martín Santamaría. Sonsoles, when you want, you can introduce the speaker today.
Thank you, Monica. Yes, welcome everybody. We have today Professor Francisco Corzana to deliver this seminar entitled Synthetic Glycopeptides, Advancing Cancer Vaccines and Detection. Dr. Corzana is a full professor of chemistry at the University of La Rioja and the group leader of the Chemical Biology group at the University of La Rioja. His research group focuses on the synthesis and structure of glycopeptides for biomedical applications. He earned his PhD from the University of La Rioja with a dissertation on the preparation of artificial amino acids. He then pursued two postdoctoral positions, one at the University of Copenhagen with Professor Engelsen for more than 2 years, where he investigated the conformational behavior of starch-derived carbohydrates using molecular dynamic simulations, and another, a second postdoctoral stage at the Instituto de Química Orgánica General from the CSIC, from the Spanish Research Council in Madrid, with Professor Jiménez Barbero and Dr. Asensio, where he prepared modified aminoglycoside antibiotics and studied their conformational behavior in water using NMR and molecular dynamics simulations. In 2005, he returned to the University of La Rioja as Ramón y Cajal researcher and became associate professor in 2008. His current research interests include the design and synthesis of glycopeptides to develop cancer vaccines and to detect cancer and at early stages of the disease. He has authored more than 160 articles in high-impact journals and has delivered many invited talks at national and international conferences. Among the different awards and honors he has, recently Professor Corzana received the prestigious Medalla José Barluenga by the Organic Chemistry Group from the Spanish Royal Society of Chemistry, recognizing his scientific career after the initial stage of independent research, where he has demonstrated a significant leap in quality and excellence. Well, thank you, Francisco. Well, thank you, Paco, for accepting the invitation to deliver this seminar. I hope it will be very interesting for the members of the Society of Biochemistry and Molecular Biology of Spain, but also for other people that has joined the seminar. And well, the floor is yours.
So thank you very much, Sonsoles. First of all, I would like to thank you and also the Sociedad Española de Bioquímica y Biología Molecular for inviting me to share with you part of the research that we are doing at the University of La Rioja. So as you can see, the title is Synthetic Glycopeptides and its use for cancer vaccines and detection. So to do this, we focus on studying these molecules. All right. So these are mucins, and here you can see this is the peptide backbone of this molecule. This is an O-glycoprotein. So that means that the sugars are attached to the serine and threonine glycosidic linkages via alpha-O-glycosidic linkages, sorry. And the first sugar is always a GalNAc, N-acetylgalactosamine, which is represented here, as you can see here, with a yellow square. These molecules are involved in very important biological processes, as you can imagine. For example, they are involved in cell protection because most of them are on the surface of the cells. They are also involved in inflammation processes and cell signaling. And nowadays, they are attracting real interest in therapeutic approaches, and in particular, the vaccines for the treatment of cancer. and also different diagnostic tools. One of the most famous, most popular mucins is this one, MAG1. So here we have the tandem repeat sequence of this molecule. As you can see here, we have 20 amino acids, and there are 5 potential glycosylation sites, as you can see here, these 3 threonines and 2 serines. And why this molecule is so important? As I have said before, in a healthy cell, this molecule, which is here, is decorated with complex carbohydrates. On the contrary, on a cancer cell, this molecule is bearing very simple truncated carbohydrates. In some cases, just a GalNAc unit, as you can see here. So, as a result, different antigens that are masked here due to these huge carbohydrates are now exposed to the immune system and can interact with this immune system. So that's why we can develop vaccines. One of these antigens is this one. So this is the simplest one from a structural viewpoint. We have a GalNAc attached to a serine or threonine amino acid. And this is a tumor-associated carbohydrate antigen. There are more of these ones, such as the TN antigen. Here we have a galactose attached to this GalNAc via beta or glycosidic linkage of this oxygen-free, and also the STN antigen, for example, and here we have a unit of sialic acid that is linked to this OC of the GalNAc unit. And also it's very important because here the peptide backbone can be also recognized by different anti-MAG1 antibodies. So here now these small peptide fragments are exposed to the immune system. Here it's impossible due to these carbohydrates. So, and it is important to say that these anti-MAG1 antibodies appears at the early stage of the disease. So they can be used as biomarkers, as we will see later, to detect cancer. In the context of vaccines, it is important to say that this simple or truncated MAG1 present in cancer cell is present in around 90% of the cancers, as you can see here. So we are not going to use the vaccine just for one type of cancer. It's quite general, as you can see, universal. And these vaccines normally will be applied as a therapy in a therapeutic approach. That means that once a person is suffering from cancer, the idea is to treat the person with these vaccines, and then this vaccine will elicit antibodies, such as this one, and these antibodies will be able to link in a selective way only to cancer cells and destroy them. So in principle, we can avoid low side effects, for example, that are present in chemotherapy, because they are quite, in principle, in theory, they will be quite selective. We have to say that these vaccines work properly in mice, and they are able, as you can see in this image, they are able to reduce the tumor size. However, when we try to apply them to a human being, there are important problems. Here on this website, you can check the clinical trials with the different vaccines based on MAG1, on this glycoprotein that are being tested or that are now tested in humans. And none of them, we can say that none of them has passed the phase 3 clinical trial. And why? The problem is that this truncated MAG1 is an auto or self-antigen, so they can be suffered from immune suppression. I mean, our immune system can tolerate somehow this MAG1. So this is what we do in our group. We try to modify these natural antigens that are the O glycoproteins or MAG1. So using chemistry to obtain a natural or artificial antigens. So we can modify, for example, the amino acids, different amino acids of the peptide sequence, also the sugar moiety, or even the glycosidic linkage, as we will see later. So using these unnatural antigens, we can have higher stability in comparison to the natural ones, because in principle they are more stable to degradation of to enzymatic degradation. This is very important for the vaccine because it's going to be more time in circulating. And also they are more, in principle, they can be more immunogenic in the sense that they can be recognized easily by our immune system, saying, okay, this is an strange element. And this is our hypothesis. So in principle, in order to obtain a potent antigen with a potent immune response, we consider that these unnatural or artificial antigens must have high affinity or at least similar affinity with anti-MAC1 antibodies than the natural derivatives. This is also related with the idea that these compounds should present a similar conformation in solution that the natural MAC1. And why we consider or we think that this is important? Because we think that this is important to obtain a cross-reactivity. That means that we use these unnatural glycopeptides for the design a vaccine. This vaccine can elicit antibody, but the antibodies and these antibodies must recognize natural MAC1. And we think that this is important to mimic as much as possible the conformation of the natural one in solution and also bound to antibodies. So as you can imagine, when we prepare these unnatural glycopeptides, we always need to study the conformational behavior in solution and in the bound state. So I'm going to explain briefly the protocol that we use to study these small glycopeptides in solution. They are flexible molecules and they are very difficult to study by NMR because, as you know, all the NMR data that we obtain are an average over all the possible conformation. So we, first of all, we need to synthesize the compound. Then we perform different NMR experiments, for example, proton-proton distances, so we can obtain relevant distances for the conformational analysis using these NOE experiments, and also coupling constants that, as you know, are related with the torsional angles. And then we use an approach that we call molecular dynamic simulation with time average restraints. So basically, that means that we can obtain, in the end of the day, a structural ensemble, such as this one, that is able to reproduce the experimental data. Imagine that we have different frames of movie, so these are the superposition of these different frames. So this is the protocol that we normally use. And now I'm going to explain also briefly the conformational behavior of this MAC1 in solution. So here we have this structural ensemble of a MAC1 glycoprotein. So this is the peptide backbone, and as you can see, it's more or less present an extended conformation. And here we have the sugars. Some time ago, Professor Daniszewski and co-workers said that this extended conformation is due to a hydrogen bond between this NH and this carbonyl group of the serine or threonine amino acid. However, if we take a look at this distance and also at this angle, these values are not expected for a conventional hydrogen bond. So that's why we decided to study some time ago, in 2006, 2007, the conformational preferences of the TN antigen with serine on the left and threonine on the right using our protocol. So as you can see here, we have the structural ensembles of both molecules and they present a different behavior in water. Indeed, in this case, we have the GalNAc and the peptide backbone that present an almost parallel orientation. However, in the case of the threonine, as you can see, the sugar is almost perpendicular to the peptide backbone. This is due to a different glycosidic linkage conformation for both molecules. And this conformer, in the case of the threonine, can be detected by this NOE cross-peak using NMR. Also, as you can see in this structural ensemble, the serine derivative is more flexible, which is also in line with this higher value for the coupling constant. And this is important because instead of this hydrogen bond proposed by these authors, we propose bridging water molecules that connect the sugar moiety with the peptide backbone. So we think that the water molecules play a key role in the conformational preferences of these two molecules that are very important for the preparation of the vaccines. Indeed, in 2018, we were able to determine the conformational preferences of these two molecules, but in gas phase experimentally using a protocol that has been developed by Emilio Cosinero, and is we can put these molecules in the gas phase, which is very difficult, as you can imagine, and then obtain the high resolution infrared. And if we compare the the experimental with the theoretical one, and this is and the conformation that are able to reproduce this infrared spectrum, you can observe that both molecules, this is the molecule with serine and the molecule with threonine, present exactly the same conformation in the gas phase. So that means indeed that water plays a key role. And we could also obtain the crystal structure of one of the anti-MAC1 antibodies, SM3, which is probably one of the most famous anti-MAC1 antibodies, with this unnatural glycopeptide. So here you can see a fluorine atom, which is here, and we could trap in the crystal structure the water molecule that is exactly in the same place as predicted by our calculations. Obviously, we can also obtain the structure of the natural glycopeptide bound to this to the SM3 antibody. So here we have this antibody. This is the epitope for the antibody. So in yellow we have the peptide backbone. In blue we have the GalNAc, and in purple we have the amino acids of the antibody that are interacting directly with this antibody, with this antigen. Sorry. As you can see, the sugar here we have O6 is interacting through a hydrogen bond with this tyrosine, and also we have a very nice CH pi interaction with this tryptophan here. So we could explain in 2015 why these antibodies prefer the binding to the glycosylated compounds, because indeed this sugar is interacting directly with the antibody. So if we put all these pieces together, we can design different vaccines. So so far we have designed 4 different vaccines, as I'm going to show you on this slide. So we normally use the tandem repeat sequence of MAC1, and we normally modify this threonine here, which is glycosylated with the GalNAc. So for example, in this case, in these 2 cases, we have modified the threonine we have replaced the threonine by 2 different unnatural amino acids. This is the alpha-methylserine, and here we have a hydroxy norvaline amino acid. So here, instead of having a methyl group, we have an ethyl group. We can also modify, for example, the sugar, and here we have an sp2 iminosugar, and we have even modified the glycosidic linkage, such as in this case, instead of having the oxygen or the O-glycosidic linkage, we have put here a sulfur atom. And also in this particular case, we have modified this proline here, this one, by this fluoroproline. So in order to explain the rational design behind this vaccine, I'm going to explain in detail this vaccine. So as I have explained before, these molecules, this MAC1, present an extended conformation in solution, as you can see here, mediated by bridging water molecules, as we have suggested. However, in the bound state with SM3 antibody, we have a folded conformation, as you can see here. So there must be an enthalpy-entropy compensation mechanism. And indeed, we have explained before that this GalNAc is interacting directly with the SM3. So we thought, okay, if we replace this oxygen here by a sulfur oxyselenium atom with a higher, let's say larger atoms, we could increase the distance between the GalNAc and the peptide backbone. So in that case, we can modulate these interactions between the sugar and the peptide, and we could have in solution also this folded conformation in order to obtain a glycopeptide with high affinity. So this is what we did some time ago. So we synthesized these 3 glycopeptides here with oxygen, sulfur, oxyselenium, and using our approach, these are the different structural ensembles that we obtain. So this is the natural glycopeptide. So we have here the peptide backbone in green. In red, we have the sugar, the GalNAc. And in purple, here we have the conformation of the peptide, but in the bound state with SM3. As you can see here, we have more or less an extended conformation, and in these 2 cases, with the sulfur or selenium atoms, we have this folded conformation that matches quite well the conformation observed in the bound state. We determine using SPR the KD, and as you can see, the unnatural derivatives, these two, present a similar or slightly better affinity than the natural one. So this is important for the design of the vaccine. And which is also important is that, as you can see, this KD was determined at two different temperatures. In the case of the unnatural derivatives, the KD is similar at the two temperatures. However, for the natural one, we have an important difference between the values at the different temperatures. So that means that indeed we have this enthalpy-entropy compensation that we have explained before. On the other hand, this is the crystal structure of the natural glycopeptide with the SN3. And now I would like to focus on this proline amino acid. As you can see here, we have a very nice interaction, CH-pi interaction with this tryptophan. So again, if we want to obtain a natural derivative with high affinity, we could increase this interaction. And how can we do that? Using chemistry, okay? We can, for example, polarize these bonds here that are interacting directly with the aromatic part of the molecule. So this is very easy if we put, for example, a fluorine atom here. So this is what we did. And these are the affinities using biolayer interferometry. As you can see, the fluorinated derivative present the highest affinity. So now we have a very nice unnatural glycopeptide that at least present a similar or even a slightly better affinity than the natural one. So we conjugated this small glycopeptide to gold nanoparticles because they present low toxicity in principle, and this is important, they allow us to have the antigen in a multivalent presentation, which is important also for the immune response. And then we perform an immunization campaign using this protocol that you can see here. And to make a long story short, I can say that we obtain very high values of IgG antibodies in comparison to the negative control, which is just a PBS buffer solution, as you can see here. We obtain mainly IgG1 antibodies and a very low amount of IgM antibodies. That means that this T cell dependent immune response has been activated. And which is very important for us, that corroborates our hypothesis, is that if we compare this vaccine with the natural one, that present here proline, instead this fluoroproline, and here threonine with a typical O-glycosidyl linkage instead of this S-glycosidyl linkage, as you can see here, the number of antibodies is lower in comparison with our unnatural vaccine, which is so this result is interesting. Also, it's very important to determine that the antibodies elicited by this vaccine using this artificial antigen is able to recognize human cancer cells that express on the surface this truncated MAC1. So we use flow cytometry and these two cell lines that express MAC1 and this cell line as a negative control. And as you can see, the antibodies elicited in mice by this vaccine, by the natural one, is able to recognize natural MAC1. And also we use confocal microscopy, and again, is the same. So basically, if we see the green color on the surface of the cell, that means that our primary antibody, which is the antibodies elicited by the immune system of the mice, is able to recognize these two cancer cell lines. We were able also to test the antibodies with in a real context with biopsies from breast cancer patients and healthy volunteers. And as you can see here, only the antibodies developed by the vaccine are able to recognize these cancer cells and not the healthy ones. So this is quite promising in result. Okay, so now, as I had said, the idea was to use this unnatural glycopeptide to prepare this heterogeneous vaccine. That means that we have this gold nanoparticle and then we have lots of copies of this MAC1. However, now we think that it could be interesting to develop an homogeneous vaccine. So that means that we can have only one copy of the antigen. So in that case, we think that we can modify this protein, this non-toxic mutant of diphtheria toxin that has been used in human beings for the preparation of different vaccines, such as this one. So the idea is to have just one copy, because we have realized that, for example, when this protein is modified with a lot of copies, then the carrier protein, this one, has a very low immunogenicity. So this is what we did, and we have modified this protein just one copy. Just we have two different disulfur bonds, as you can see here, and we have reduced one of them, the one that is exposed to the solvent, using the conditions shown here. So we have installed this acido group, and then it's very easy to put, this is our natural glycopeptide, and using this scaffold, it's easy to perform a click chemistry. So we have just our protein, carrier protein, with one copy of the antigen. This is the MS expected for this compound, and here we have a model of the vaccine. So we have the carrier protein, and here just one copy of this MAC1, which is exposed to the solvent. Okay, and again we have performed an immunization campaign using this protocol, and you can see here that this in red we have the vaccine, and these are negative controls. We can increase the antibodies that are elicited after, for example, the 4 injection. We obtain in this case IgG2 instead of IgG1 with the heterogeneous vaccine. We also check that the vaccine is able, that the antibodies are able to bind to this cancer cell, human cancer cell that's expressed MAC1 on the surface using flow cytometry. And again, using, for example, confocal microscopy, we can obtain a similar result as previously obtained with the vaccine with the cold nanoparticles. Then we have checked this vaccine as a prophylactic vaccine. So that means that so we have these mice have been treated with the vaccine, and then tumors were induced 6 days after the last injection. In this case, we have used this colon adenocarcinoma that has been modified to express MAC1 on the surface. And again, these are we check every 3 days, as you can see here, the tumor volume, and as you can see, this tumor volume is the growth is reduced when we use the vaccine. Remember that this is the red color, and this is the PBS, and this is just the carrier protein. And here we have the tumor weight at day 15, for example, and again, using the vaccine, you can see that we have stopped somehow the growth of the tumor. And also, for example, we perform this cytotoxicity assay using the CD4 and CD8 plus T cells from the mice spleens. And you can see that, for example, the viability of these cancer cells is reduced when we use the vaccine. Okay, so this is us using the vaccine as a prophylactic approach, but we have used also as a therapeutic approach of the vaccine. So first of all, we inoculate the tumor, and then we treat the mice with the vaccine every 2 days. So in this case, we have used the same tumors as in the previous case, this colon adenocarcinoma, and also a pancreatic model. And to make a long story short, you have to say that, as you can see here, we can reduce the growth of the tumor in both cases, and in particular for the pancreatic cancer, and also that we can increase the probability of survival in both in both cases, and again in particular in the case of the pancreatic cancer. So these are very promising results. And also we have used as a therapeutic vaccine, and in this case in combination with an immune checkpoint inhibitor. So first we inoculate the tumor, and then we put our vaccine every 2 days, and every 3 days we add this checkpoint inhibitor, which in this case is an anti-PD-1 antibody. So as you know, the cancer cell can inhibit somehow the T cells by developing this complex. So if we can destroy this complex using, for example, a checkpoint inhibitor, then we can activate these T cells. So this is what we did. And again, for these two cancer cell lines, for the colon and pancreatic cancer, these are the results that we obtain. In this case, we can increase the probability of survival substantially, as you can see here, and in particular for this colon adenocarcinoma. So these are promising results, and now we are developing more tests in trying to increase the dose, just to see if we can increase also the probability of survival and also reduce the growth of the tumor. And now I'm going to move to the second part of the talk. So as I have said before, we use these unnatural glycopeptides for developing of vaccines, but also to detect cancer. So just some statistics here on this slide. So for this year, these are the estimated new cases for male and female cancer. For female, the most important one, as we know, is prostate, and for females, breast cancer. And I want to show you pancreatic cancer is the number of cases is not too high in comparison with prostate or breast cancer. However, the estimated deaths is quite high. And this is because it's very difficult. This type of cancer, pancreatic cancer, is very difficult to detect. So then it's very difficult to treat. So we are trying to develop a system to detect this pancreatic cancer at early stage of the disease. So as I had said before, cancer cells present on their surface this tumor-associated MAC1. So these molecules can be detected from the surface of the cell and go into the bloodstream. So in principle, we could use this tumor-associated MAC1 as biomarker using a test to detect it. However, the systems that have been developed so far present low sensitivity and low specificity. And there is a different approach in which we are working on, which is as we had said, there are different studies that indicate that patients that are suffering from cancer develop at the early stage of the disease anti-MAC1 antibodies. So we could use our antigens to fish out these antibodies. So in principle, they can be detected. We can detect cancer at early stage of the disease. However, the problem is that the concentration of these antibodies is very low. So we need to have antigens with a very high affinity to be able to fish out as much as possible, as much antibodies as possible. So again, using chemistry, this is what we are doing. We are trying to obtain unnatural or artificial antigens that are very good, let's say, fishing hooks. So we can, in principle, we need to enhance antigen-antibody affinity, and probably we will have, in that case, a system with high sensitivity and high specificity. So in this context, and in 2020, we were able to determine the crystal structure of antibody 5, which is able to recognize this sequence of MAC1 and also with the GalNAc. This antibody has been used, for example, to treat pancreatic cancer using the CAR T-cell technology, and it has been quite successful, the treatment. And this antibody is quite peculiar, as you can see here, because the main epitope of the antibody is just the GalNAc unit. As you can see here, we have different hydrogen bonds and also CH-pi interactions. So today, I'm going to focus just on this interaction. We have here a methyl group of GalNAc and this histidine, and we have a very nice CH-pi interaction. So we thought, okay, if we want to increase the affinity, we can enhance this interaction, replacing, for example, this methyl group by an ethyl group. So in that case, we can have the methyl group close to the to this aromatic ring. So this is what we did. We prepared 3 different compounds. As you can see, we have the epitope recognized by the antibody with GalNAc. So we have the natural compound, compound 1. We put here a fluorinated derivative to use the same approach as previously used for the fluoroproline in order to polarize this CH bond and increase the CH pi interaction. And also this one, in which we have replaced the methyl group by this ethyl group. And we have performed the SPR assays to determine the affinity. And as you can see, compound 3 presents a high affinity in comparison to the natural one. And here we have, for example, the molecular dynamic simulation in which we have, this is the ethyl group, which has a very nice CH pi interaction with this histidine 50. We have recently obtained this the thermodynamic parameters. So as you can see, the case of the unnatural one, or in both cases, we can say that the enthalpy is the driving force of the binding, and in particular for this one, for the unnatural one. So indeed, probably we have enhanced this CH pi interaction. Then, with these compounds in our hand, we have put these molecules, we have conjugated to gold nanoparticles in order to have better reproducibility for our system, and we have done a classical dot blot analysis. So with the nitrocellulose membrane, so we have put we can use for the setup a commercially available 5/5 antibody, or also serum from the patients, and then a secondary antibody for the readout. And these are the values that we obtain. For example, this is for glycopeptide 1, the natural one, the natural one with fluorine atom, and the one with the anethyl group. And as you can see, we can using 5/5 antibody, we can detect very low amount of this antibody, and in particular with this compound number 3. So then we use real samples. So we could obtain 20 samples from patients with pancreatic cancer and 20 samples from healthy volunteers, and we perform this assay with compound 3, and as you can see, there is a clear difference between a patient and healthy volunteers, and also the levels of the antibodies, the MAC1 antibodies, is lower in the case of the control in comparison with the patient. And what is more important is that our system, which is here, this one red, present a higher sensitivity and higher specificity than the 2 ones, this one and this one, that are nowadays used in clinical for detecting this type of cancer. So now we have obtained a proof of concept in order to develop a protocol to use directly in a clinical using the serum of the different patients. And we have also a pattern with this process. And this slide for me is very interesting because in the context of trying to fish out these antibodies, because we have developed different glycopeptides that present the 5/5 epitope as the ones as I have shown before, other glycopeptide with SN3 epitope, and then the full length tandem repeat with both epitopes present on the same molecule. And we have, using a rational design, a structure-based design, we have prepared different glycopeptides with very high affinity for 5/5 antibody and also for SN3 antibody. So this is compound number 3, and in this case, in red, we have a positive case. That means a sample from a patient that is suffering from cancer, pancreatic cancer, and the blue is the negative control for healthy volunteer. And this one, compound number 3, is able to discriminate between positive and negative cases. However, for example, compound 4, that has been designed to present a very bad affinity for the antibody 5/5, is not able, this one, as you can see here, is not able to discriminate between positive and negative cases. But for me, the most important thing is this one. As you can see here, for example, compound number 9 presents a very high affinity for SN3 for antibody SN3. However, is not able to discriminate between positive and negative cases. So that means that at least for pancreatic cancer, we need to have antigens that present the 5-5 epitope. And maybe this is also related with the use of 5-5 for the treatment of pancreatic cancer using the T-cell technology. And also in this case, for the full-length tandem repeat, only in these two cases, when we compound 10 and compound 12, in which we have the epitope of 5-5 antibody, 10 and 12, only these two systems are able to discriminate between positive and negative cases. So I think this slide is also very important because that is not only is not necessary or it is mandatory to have a very high affinity to fish out these antibodies present in cancer patients, but at the same time, it is important to have the correct, let's say, epitope. And in the case of pancreatic cancer, apparently the 5-5 is required. So this is all the talk. So finally, I would like to thank all the people of my group. Obviously, this is impossible that I can perform all this experiment. So thanks to all my group, and especially to the students that have been involved in these projects, and also our collaborators. As you can imagine, we need to collaborate with a lot of people because this is a multidisciplinary approach that we are using, so it's impossible to have all the techniques in our institution. And obviously, I would like to thank also these institutions for financial support, in particular Universidad de La Rioja and Asociación Española contra el Cáncer, and all of you for your attention. So thank you very much. So, yeah.
Thank you very much.
So I can. Oh, sorry. I don't
Thank
know.
you very much, Paco, for this impressive work and very multidisciplinary work. Yes. Well, I forgot to tell the people that are attending that they can write questions either in the chat, whether if they are registered in the Zoom session, or either by the YouTube streaming. They can also send questions to our speaker. So, well, while we are getting questions from the people that are attending, I have some questions myself.
Okay.
And maybe Monica also has some questions. So, well, I'm going to start with one, and then, well, regarding the design of these GalNAc derivatives, I have several questions. One is, given that these conformations that are involved in a water molecule that is bridging this conformation, this was on. Can this give you ideas about the design of maybe rigid derivatives, cyclic derivatives? Yeah. Because I've seen that you have one of the modifications that you have proposed is this kind of with a
cyclic.
Yes, but not in the other region of the molecule. So
you are right. Yeah, and this is a very good question indeed. We are trying to do that. It's not easy, but we are trying to connect directly the N-acetyl group with the peptide backbone. Yeah, this is a very nice idea indeed, and we are now working on that point. Yeah.
I guess it must be difficult from the synthetic point of view, and probably...
Yes, it's not easy, and so far we haven't obtained the compound that we want to obtain. I mean, it's not easy because there are...
Okay. So you already have some designed molecules,
yes,
because
yes,
also
yes.
there are chiral...
We are working on that,
yeah.
But it's very interesting indeed, yeah. This is our
very
next...
nice.
Yes.
Oh, yeah, your next goal.
And in particular for trying to obtain a good system for detecting cancer. Because as I had said, in the case of the vaccine, I think that is enough to have similar affinity, similar conformation of behavior. But in the case of detecting these antibodies, you need to have a very high affinity. And probably using this approach is possible to increase the affinity substantially.
Regarding the application as vaccines, you said that they are able to, in principle, to be applied for a very wide variety of cancers. Have you already tested this in other types of tumors?
So far, we have tested these two tumors, this colon adenocarcinoma and pancreatic cancer models. They are from mice, but they are designed to express MAC1, human MAC1, but only these two so far. But I think that there are some authors that have been tested different types of cancer, and I think in particular the pancreatic cancer, which is one of the most difficult to treat nowadays, and there are a lot of efforts in this direction. Yeah. So.
Well, we already have
related to this that Sonsoles was asking you. So one idea would be to check how many or which other cancers can have a high expression of mucins, no?
Yes. Yeah, but in general,
how do I guess breast cancer has also.
Breast cancer, yes, you are right. Yeah, we are now trying to test the vaccine with this cancer, this cancer model. Yeah, breast cancer, and also ovarian, and yeah, these are the maybe most typical ones. Yeah, and for sure that they express this tumor-associated MAC1 on the surface. Yeah, and also we are trying to detect this type of cancer because of that. So it's more or less the same. Yeah.
You do both, I guess, in parallel.
Yes,
as soon as you have the molecules, you
in parallel. Yes. Probably it's easier to use this antigen to detect cancer because once you have the samples, it's very easy to check. But in the case of the vaccines, I think it's very difficult, to perform the in vivo experiments. It's not easy, you need to have a lot of mice, and sometimes some of them death suddenly, so it's not easy. Yeah.
Complicated.
Yeah.
One thing that I didn't get about the first part, that your mice are immunocompetent or they are in your models?
No, in principle, they have to produce antibodies.
Yeah, so they are fully immunocompetent.
Yeah.
Okay, but the cancer cells that you inject, they are not orthotopic.
They are cancer cells from mice, I mean. But that have been engineered to express on the surface MAC1, this tumor-associated MAC1. Yeah. But this is also a good point. Yeah, we need to do that. Yeah. Otherwise, yeah.
Okay, thanks.
We have some questions already from the audience. The first one is from an anonymous participant that says, hi, why is using the TAR, the time average restraint approach in molecular dynamics important and practical for your workflow? Isn't this limiting the example being sample, the ensemble being sample?
Yeah. No, this is also a very good question. When you have a very small, when you have a protein, it's very normally you have a main conformation, which is very easy using classical restraints from the NMR. You obtain the molecule just with the main conformer. But in the case of flexible small glycopeptides, they present different conformer in solution. So once you determine NOE, for example, the distances are an average over all the possible conformers present in solution. So that's why you cannot use the classical restraints. If you use classical restraints, you normally obtain an artifact conformer, which is an average of all the possible conformation. Using our approach, the idea is that the restraints can be satisfied in average, but not for a specific frame. That means you can have, for example, for one step is not satisfied this restraint, but in average you need to satisfy. So you can, in this way, you can avoid these artifacts that you obtain using classical restraints. I mean, if you use the classical restraints, for sure that you obtain normally artifacts. So the idea is we perform the enema, we obtain distances, and these distances are used as this time average restraints in order to obtain at least a possible solution of the system. You obtain an ensemble that is able to reproduce all this experimental data. Maybe it's not the only one, but at least you obtain one of them. So, but this is important, yeah.
Thank you, Paco. Another question by Janet Sanchez. Do you check the protonation states of the histidine that you use as a reference to decrease the distance towards GalNAc?
The histidine.
The histidine, yes.
I suppose, yeah. Normally this is not protonated, but yeah, because you could expect that probably in general this histidine and according to the presentation of the histidine with the different residues that are around the histidine, this histidine is not positive charge. So this is, I don't know if this is the answer, but
or well, following this question, how do you decide which nitrogen is the one that is taking the hydrogen? How do you decide if it's the epsilon or the delta? Maybe from the structure. Or maybe it's not important. Maybe it's not important.
We normally use software to determine which one is the nitrogen. The most probably protonation state, but using, for example, amber, you use the classical HIE with the epsilon 1, with the proton in the epsilon 1, but for example, we normally use different software, Maestro, in order to predict the charge, or if one of these amino acids present the charge. But in this particular case, I think it's not the case due to the environment.
Thank you, Paco. And we have a third question from Marta Artola. Hi, Francisco, impressive work. I was wondering, did you consider to generate the trifluoracetyl derivative as well?
The trifluoracetyl, yeah.
Strength.
We have already done this one with the 3 fluorine atoms, right? And in this case, we don't obtain a beta affinity. And the problem is that if you remove all the CH bonds, then you cannot have this CH pi interaction. We have replaced by one to have 1 fluorine, 2 fluorine atoms, and in this case it works, but not in the case of 3 fluorine atoms. Yeah.
So the monofluorinated is the best one. Is
the best, yeah. Because you have a balance, you increase the polarization of the bonds that are interacting with the aromatic part, but at the same time you reduce the number of CH bonds. So you need to have a balance.
A balance.
And also it's very important because as we know, the fluorine has some peculiarities concerning the hydrophobicity. So
yes,
by this particular atom. So but we have already tested that, and the 3 fluorinated compound cannot increase the affinity.
But so there is a lot of synthesis behind your research. You've already synthesized many compounds.
Yeah, normally we put the compounds, but the synthesis of some of them is difficult. Other ones are very easy to prepare. It depends. For example, for the detection system with the M-propionyl group, it's very easy to prepare. So.
Thank you, Paco. Monica, is there any question from the YouTube?
No.
I have also a curiosity about the in the last part of your presentation that you were trying to use the vaccine as a prophylactic.
Yes.
How is the administration? Is intramuscular, intravenous? How is the administration via that is used?
I think
Because there was the drawing of an injection, so maybe it was intravenous. I was wondering about.
I think so. I think it's in this case, yeah. I haven't done the in vivo experiments, but I think it's subcutaneous. I think so. Yeah, I think so.
No, because I was wondering about the absorption properties of these compounds, the distribution, etc. Okay, so.
Yeah. But I think, indeed, I think that we have tested both, different approaches to obtain the better results. And also now we are trying to increase the dose, to see if it is toxic or not. So far, we can say that it's not toxic at high concentrations, and we can decrease the growth of the tumors. But this result was obtained yesterday, and I have to check it carefully. But indeed, yeah, I think that we can increase the dose without increase the toxicity of the vaccine, which is also very important.
Excellent. Yes.
Mhm.
Monica, do you have more questions or?
Well, I was wondering, but if you say that the results are very, very recent, I don't know. I was wondering if you have checked how the cells in the tumor die. I mean, because you see a decrease in the growth of the tumor. So I just was wondering if you know how they are. I mean, they are not growing or they are dying, or I mean, if you have any idea about the behavior or the mechanism.
I don't know if I suppose that the antibodies present cytotoxicity, I suppose. I don't know, but I mean, we need to check that carefully. Yeah. We have also checked that we have different cytokines, that have been developed, and we are trying to analyze all this data.
If it's an inflammatory death or maybe it's an apoptosis or something like that. Okay.
We are working on that, yeah, because these are very new results. Yeah.
Okay,
well,
so.
But for me, the most important thing is that there is a dogma saying that for developing of vaccines, you need to have a multi-presentation of the antigen. And with this vaccine, we have demonstrated that it can work just with a copy. So you can have, this is also important in order to prepare a batch of the vaccine to have reproducibility. For example, if you have the nanoparticles, it's difficult to obtain always the same distribution. More or less, you are going to obtain the same if you do that in the same conditions, but you are never sure that this is the case. But when you have a chemically defined vaccine, it's easier. You always obtain that vaccine. So we think it's very interesting result, because normally people use a lot of copies of the. With this protein, carrier protein, we have realized that if you put a lot of copies, then you reduce the immunogenicity of the protein, which is also important to stimulate the immune response in mice. So we are happy with this.
Very interesting.
Very interesting and impressive work, Paco. Very multidisciplinary from the design and chemical synthesis and studies of molecular recognition and to mice models. And so, well, we wish you the best, because these are very promising molecules indeed. So
thank you. Yeah, we are trying to work hard to obtain in the future vaccine. Yeah. Okay.
Thank you very much, Paco.
Thank
you also to the participants.
Yes, thank you to the audience, to Sonsoles to organize the seminar, and of course to Paco for a very nice talk.
Thank you.
So see you in the next SEM webinar, I guess. So have a nice day.
Thank you.
Thank you. Bye.
Bye.